My name is Drew Davis, and I'm a student in Bio 301 and doing a presentation over my citizen science project. For my topic, I picked the Rodent Little Brothers Secret Lives of Mice on the website Zooniverse. When looking through a topic list, I thought this one really jumped out to me immediately. Uh, there was a number of reasons that it caught my eye, and my mom is a fourth grade teacher at a private school in our hometown, and she always puts a lot of effort into teaching science. Every year, she will go up to Indianapolis and pick up two albino rats for her class to complete an experiment. It's one of the highlights of her classes here, and one of the coolest science things they do. So basically, the rats are put into separate cages, and they live on two separate diets. One drinks sugar water, and one drinks milk. They're given grains, fruits, vegetables, and meats. The students track the energy, the health of the rats, and the growth of the rats over several weeks, and then they chart their results for their experiment. And once the experiment's complete, she picks two students out of a hat, and they get to take the rats home, and then they become pets at that point. And I think that's probably another big reason that her class looks forward to it. And thought it would be something really neat I could do because it was similar, and it was the closest thing I could find on the Zooniverse website. The purpose of this project, in this experience, or excuse me, in this experiment, scientists are tracking the morning routines of a red dot mouse, the blue dot mouse, and a green dot mouse. Since 99% of human genes are predicted to have equivalent genes in mice, scientists are hoping to gain information from behaviors citizen scientists are tracking into genes that cause diseases. All three of the mice are in the same cage, as we can see below. There's a camera outside of the cage that records the mice's actions 24/7. You could see kind of in my presentation, bottom right corner, I have the cage and the diagram of the cage. You see where the camera is located. You see where the lighting's at, and you kind of see the cage structure and how they go about things. And then in the bottom left, I have a picture of kind of the live cage environment rather than the diagram. Basically, what you see when you're watching the videos, you see a little toilet paper roll they can use to play with, the water bottle, the feeding tray, and the whole area down there. This project creates up to 1 terabyte of data per week, which is a lot. The mice are all in the same cage that I mentioned, and scientists analyze the behavior and activities of each of the three mice. I spent an hour observing each one as well. And for more, if you want to learn more on that, you can kind of look at my diagram and picture of the cage if you want to see the environment they live in a little bit up closer. The next thing is, what are citizen scientists doing to help answer the question? Well, as a citizen scientist, I'm helping scientists study the genes of behavior, or excuse me, I'm helping scientists study genes of mice behavior in a home cage environment. The ability to define early disease characteristics in the home cage environment will provide scientists with vital information on the early stages of diseases and potentially how to treat them. Collecting much more data on each individual animal will reduce variation in each experiment and increase the statistical power of testing so that less animals need to be used, which obviously the less you use, the better. Citizen scientists are helping real scientists collect the data and study the genes in humans. There's a picture of the mice we are studying right there in the bottom. And I'll take you guys to the website and kind of show you some of my contributions as well. As you can see, there's collections here. Everyone that's participated kind of goes in, and then there's three little bars at the bottom of each video you watch, and you can go in after you watch a video. And I thought this was neat. You can classify it and group it. So like one of my things I was tracking in my data was climbing, and obviously another thing was eating. So as I went through and was making my tally marks, not only was I doing that, I was also grouping that data into a specific set for other people to go back and analyze, or maybe even I want to go back and analyze it, I could. So you could see for this one, there's an example of all of the data I collected in there, and that was just a little bit that I did along the way. And I would kind of like, I felt like if you labeled them, it kind of made it easier for everyone else that was participating to kind of see what you were doing, and that's how I was kind of contributing. Let me go back to my PowerPoint. As I mentioned, I spent 1 hour studying the red dot mouse, another hour studying the green dot mouse, and another hour studying the blue dot mouse. I did these in 3 separate settings, probably over the course of maybe like a month and a half or so. I did the first one probably in like August or September, if I remember right, and the next two were closer together in like the last 4 weeks or so. I watched a bunch of short video clips that displayed the mouse's activity from various times. I tracked the activities of the mice by keeping tallies on what activity it was doing. I was able to use folder and labeling features, I mentioned, to archive the videos I was watching by activity, which I thought was a very, very useful thing. The following activities were tracked: walking, eating, climbing, drinking, grooming, and sleeping. And as I take you to my next slide, you'll be able to see the data I collected. I broke it down into hours 1, 2, and 3. I spent my first hour studying the red dot mouse, my second hour studying the green dot mouse, and my third hour studying the blue dot mouse. As you could see, there was a little bit of discrepancies in how the data differed per each mouse. As you could see, the blue dot mouse a little more active when it came to walking, whereas the red dot mouse is more active when it came to eating, and so on. As you could see across the course of my data there. I'll just kind of move on from that, and if you want to reference that later on, you can. So the next question is, based on the data I collected, what answers scientists might be able to get to the question they're trying to answer? Based on the data I collected, the mice were more active with climbing, eating, and grooming. Collecting much more data on each individual animal will reduce variation in each experiment and increase the statistical power of testing, so the less animals need to be used, I mentioned, and that's a very good thing. This project provides an opportunity to gain new knowledge about how mice behave, particularly in the night when they are most active and over much longer periods, which you would have missed if traditional tests were used. It's also possible to observe social interactions in much more detail than was previously possible with the citizen scientists collecting data. They use citizen scientists like me or one of the other people that are in the class to recognize the behaviors in the video, because computers cannot do this job. However, using lots of examples from people and combining these techniques called machine learning, they can effectively train computers to recognize behaviors from the video. Computers can learn to do this task automatically by looking at examples from us humans as we go through and kind of label and collect our data. And just discuss what questions I have developed of a topic, and based on the results I collected participating in the project. I thought it was very interesting the use of machine learning. So I wanted to know how do they train the computers using the algorithm to recognize these behaviors in mice. I like to know that, and I think that'd be kind of fascinating. The red dot mouse seemed to be more active in eating than the other two mice. Were all mice genetically the same and from the same litter at the beginning of the project? That's something I'd like to know. I think that would kind of be applicable there. And how does the unidentifiable behaviors get figured into the research? What do they do with this information, and how do they weed it out, I guess? And then, this project kind of helped me expand my appreciation of science. Prior to this project, I had never heard of doing research for a science project without having everything right there in front of me. I thought it was very neat to be able to watch videos online and kind of contribute to an experiment as I went along. During this project, I gained more of an appreciation for the little things in the scientific process. I had no clue that approximately 99% of human genes are predicted to have an equivalent gene in mice. Humans have 20,000 genes, and the function of many of these genes is unknown. Many diseases occur when there is error in a gene. By studying mice, hopefully they could find more information about these diseases and get a cure to them. I think that would be a big step in the right direction. But overall, I kind of really enjoyed this project. I thought I learned a lot, and I thought that it helped my appreciation for their science grow a little bit. And I thought by picking a project that I enjoyed, it definitely helped me be a little more engaged.